Hello friends, again welcome to my channel. Today we are going to discuss about the regulation of lactose operon. In the previous lecture we are already discussing about the regulation of arabinose operon. Now similar to that of the arabinose operon, this lactose operon, it is also a degradative operon, that is within which there is the utilization of the lactose as an carbon and energy source. This lactose operon, it is a classical example of epigenetic regulation of the phenotypic heterogeneity which is preferentially observed within the Escherichia coli. This operon model we have studied within the Escherichia coli, which can utilize the lactose as a sole source of carbon and energy. Now this lac operon, similar to the other types of the operon, it is also consists of a cluster of genes which preferentially includes the regulatory gene, promoter gene and the structural genes. The lack of majorly within the structural genes, there are the three different structural genes. These are observed within the lactose operon, that is the lac Z, lac Y and the lac A. Out of which the lac Z, it is responsible to encode the enzyme beta galactosidase, which is responsible to break down the disaccharide sugar, that is the lactose, into glucose and galactose. The second one, it is the lac Y, which is responsible to encode the enzyme galactoside permease, which acts as a transporter protein and facilitates the entry of lactose inside the cell. And the third one, it is the lac A. It encodes the enzyme beta galactoside transacetylase. It is responsible for the modification of the toxic form of the galactoside by transferring an acetyl group from acetyl CoA to the beta galactosides and remove it out of the cell to remove the toxicity. Now, majorly here it is the construction of this lactose operon. That is, whenever there is the absence of the lactose, which is acts as an inducer within the lactose operon, as similar to the arabinose operon within the arabinose operon also, or we can say that within the degradative operon, majorly the substrate it is acts as an inducer. Whenever there is the absence of inducer, that is the absence of lactose within this, at that time there is no expression of the lac operon. That means the structural genes, this cannot be expressed in the absence of the lactose, because preferentially in that case the regulatory gene, it is responsible to synthesize the repressor protein which binds with the operator site, which blocks the binding of the RNA polymerase to the operator site, and there is no expression of the structural genes. In reverse to that, whenever there is the presence of the inducer, that is the lactose, at that time this lactose, it is acts as an activator or it is an inducer. So this inducer, it binds with the repressor protein synthesized by the regulatory gene, and it is responsible to convert the active form of the repressor into the inactive form by the conformational changes within the repressor, due to which it cannot bind with the operator site. And now here there is the available site for the RNA polymerase to bind with the operator site, and it can promote the expression of the structural genes. Now the regulatory gene, it includes the lac I along with the promoter and operator sequences. The lac I, it encodes the repressor protein which is responsible to bind with the operator site. The promoter site, it is the binding site for the RNA polymerase, and the operator, it is the binding site for the repressor protein. Now preferentially the lac operon, it can be regulated by the different mechanism. The first mechanism, it is the repression, which is observed in the absence of the lactose. Now the repressor gene, it is encodes the repressor protein and which are having the capacity to bind with the operator site. The repressor protein, it is an tetramer which is transcribed from the lac I gene by its own promoter, which is independent of the lactose operon. Now this repressor protein, it can bind to the region O1 of the operator, the lac operon. Two secondary binding site for the repressor protein, it is the region O2 and which is near to the lac Z, and the region O3 which is the upstream to the promoter site. So whenever there is the binding of the repressor protein with the region O1 of the operator site and one of the secondary region, then due to the binding of this repressor, it is responsible to block the binding site for the RNA polymerase enzyme, and due to which there is no binding of the RNA polymerase enzyme whenever there is the binding of this repressor protein with the operator site. Whenever there is the immediately addition of the lactose within the media, it is responsible to induce the synthesis of these structural genes or the expression of these structural genes. But even in the repressed state, some E. coli cells, these have the minimal level of beta galactosidase and the galactoside permease, and this basal level it is essential for the regulation of the operon in the presence of the lactose. Now here in that figure we can observe whenever there is absence of the lactose, at that time the repressor protein which is present within the active state, it binds with the operator site which is near to the laxation structural gene and the promoter site which is the binding site for the RNA polymerase enzyme. So these structures, these are overlap in between these. Due to that, due to the binding of the repressor protein, there is no binding of the RNA polymerase and there is no expression of the structural genes. Now the second mechanism, it is the induction, which is observed in the presence of lactose. Now whenever there is the lactose it is present and it is acts as an inducer of the lactose operon, it binds with the repressor. Means in the presence of the lactose also there is the synthesis of the repressor protein, but in that case this lactose which is present and acts as an inducer, it binds with the repressor protein and it induces the conformational changes within the repressor protein, due to which it cannot bind with the operator site, and there is the removal of the repressor protein from the operator site, and now the site it is available for the binding of RNA polymerase to continue the expression of the genes. Now actually within the lactose operon, the allolactose it is acts as a actual inducer of the lactose operon. Whenever within the previously cases there is the presence of the lactose, some amount of the lactose during the utilization of lactose it is get converted within the allolactose and it is actually acts as a inducer of the lactose operon. The beta galactosidase it converts the lactose into the allolactose that binds to a repressor protein and inactivate the repressor protein. Now here within this figure we can observe the induction of the lactose operon due to the presence of the allolactose which is acts as an inducer which convert the active form of repressor protein into the inactive form, due to which it is releases from the operator site and the RNA polymerase it can transcribe the LAC-ZYA mRNA. Now the another mechanism for the regulation of lactose operon, it is the catabolic repression, within which can be observed whenever there is the presence of both glucose and lactose within the media, and in that condition there is the growth of E. coli cells. Now majorly such a condition it is regulated by the CAP, that is the catabolite activator protein, which is encoded by the CAP gene which is located within the another site within the E. coli chromosome. Whenever the CAP it binds near to the promoter upstream to the LAC-I, it facilitates the binding of RNA polymerase to the promoter more tightly so that the rate of transcription it increases 20-fold. Means it is an activator of the transcription process. But initially this CAP protein it is present within the inactive form. The CAMP it is required for the activation of this CAP protein. The CAMP it is a secondary messenger which binds with the CAP only in the absence of glucose. Means whenever there is the absence of glucose, there is the increased concentration of CAMP. Means this concentration of glucose and the concentration of CAMP, these are inversely proportional to each other. If the glucose it is low, then the higher amount of the CAMP it is present. Whenever glucose it is high, there is the low concentration of the CAMP. So whenever the level of glucose it is high in the cells, the synthesis of CAMP it is get inhibited. Furthermore, the glucose also transport the CAMP out of the cells and decreasing the concentration of CAMP. So in the presence of glucose, there is no activation of the lactose operon. Whenever there is the low concentration of glucose, at that time the CAMP it is synthesized within the cell, and at that conditions there is the activation of the lactose operon. Here in this figure we can observe the catabolite repression mechanism. The CAMP it binds with the CAP, it activates the CAP site and it is responsible to increase the rate of transcription. Now the another last mechanism for the regulation of the lactose operon, it is the feedback regulation. The active transport of lactose by the permeates inside the cell, it causes the accumulation of higher inducer concentration, which is resulting in the increases the expression of lac operon. Now, due to this, again, the transgalactosylation of the lactose by the beta galactoside produces allolactose and again causing the induction of lac operon. The beta galactosidase, it hydrolyzes the lactose and allolactose to produce the glucose and galactose, increases the concentration of glucose and it causes the catabolite preparation. Means the higher the concentration of lactose, no doubt the allolactose or the lactose, it is acts as an inducer of lactoperon. But whenever there is the increased concentration of this lactose, it is also responsible to inhibit its the activation of this lactose operon due to the accumulation of the glucose within the bacterial cells. So here there are the different mechanism for the regulation of lactose operon. So if you like this video, please subscribe my channel and also share this video with your friend. Thank you.